to confess, when I was asked to do this talk today, I was given the remit of coming up with a talk of combining AI, neuroscience, and Christmas. So this really put me out of my comfort zone. So I originally started with the title of AI Tools for Drug Discovery and Literature Research, which is kind of really the focus of my work at Biorelate. Although something really strange happened in the last week. Santa Claus, who was embarking on a routine training exercise with his reindeer, he noticed that they were rather unwell.
Dan, sorry to interrupt. Just to check that we're seeing the slide preview mode. We can see the slides are right. Okay, that's better.
Okay. Are you now seeing a full screen image of Santa and his ill-looking reindeer?
We're seeing part of that, but we're seeing also the slide preview mode, so next slide, etc.
Okay.
I mean, it works all right. If you want to carry on that, we can still see the slide you're talking about.
Let me
just in case you were planning animations or something.
Yeah, I've got two screens. This is the problem. So is that better now?
Yes.
Okay, right,
Thank
good.
you.
Yeah, so unfortunately for Santa and his reindeer, the vet told him that they were likely suffering from a nasty disease called chronic wasting disease, which is contagious and fatal neurological disease, which is found in all sorts of different cervids, including reindeer, symptoms of which involve hypersalivation, significant weight loss, ataxia, caused by the misfolding of prion protein. And most importantly for this particular disease is that there are currently no curative or prophylactic treatment available. So Santa was in a bit of a quandary, and he asked Biorelate if they could help him understand the current research on CWD so that him and his elves could potentially find a cure. Now back to reality here. So essentially, this is a common problem that we, as researchers trying to develop new drugs, face. We've got a really, really important disease which has potentially a very large unmet medical need, or in this case, massive ramifications to the occurrence of Christmas. And in order for us to properly understand the state of research, we have to trawl through millions and millions of different articles. So if we're looking in, the literature, clinical trials, patents, et cetera, there are something like 100 million potential articles out there in the public domain, which we can go out there and read. Most of us are able to see all of this, but to actually understand it, to properly structure the data that's contained within it, connect it, and really analyze all of this as a kind of sum is incredibly difficult. And so the way that we've typically approached this task for many, many years is to get people to read through these documents and put all this information into a database, which obviously costs money. So if you take a person to read through one of these articles, it's about $220. So, you were to go out there and capture all of the data that's stored in that text, this would be an impractical task costing billions of dollars. And of course, it's growing all of the time. Doubling every 9 years or so in terms of scientific output. So really, this is a classic task in which we can apply AI and automation to try and solve it in terms of capturing all of this data, which is out there stored in text. So the software that has to do this really has to resolve a number of different, quite difficult tasks. So I've focused on an example here where we're looking to capture a cause and effect relationship documented in an article where somebody said that the degradation of androgen receptor by MDM2 mediated ubiquitination in prostate stromal cells. And so one of the first tasks that software might have to do in this particular case is to recognize that you've got different biomedical concepts here. You've got a couple of genes and you've got a cell. And the next stage is for that software then to map these to their respective identifiers. So androgen receptor is actually the gene AR, which once you've attached the ID to that, you obviously then have all of the other information about what that particular gene is in terms of its sequence and all of the other data that you might know about AR. Same goes for MDM2 and same goes for prostate stromal cells. So the next point is you'll be looking to understand that there is some directionality between these concepts, that actually it's MDM2 which is having an impact on AR, and that because there is degradation involved here, that this is actually a decrease in either the abundance or the activity of this particular protein via the mechanism of ubiquitination. And we also have the important context that this is going on within prostate stromal cells. So altogether, the software has to get every single one of these steps right in order to capture this piece of information 100% correctly. And so in order to actually decide whether we want to use this data provided by software, it's really, really helpful if you can apply confidence metrics to this that help us see whether this is really going to be useful or not. So this is hard. And in order for software to get this right, as I say, it has to get every single one of these steps right. So what the company Biorelate does is essentially to try to do this at scale across all of the literature that's out there. So we essentially have this platform that's called Galactic AI that takes in many, many different sources of text, all of those different journals, patents, clinical trials, many of which have never even been read before because they've literally just come out that day and nobody's had the time to properly understand the data contained within it against all of the other data that's out there. And this is going on daily. And we essentially then capture all of the different types of data, which generally speaking is dark and is largely unsearchable because it's relational data, things like cause and effect, that's very, very difficult to get at from kind of general search engines. So we're using specialized deep learning software, which aims to capture all of this data as accurately as possible. And then we're supplying it to different companies who are then using this to support their drug discovery processes. So different products and services, interfaces, access to the data, as well as collaborations with different companies. So going back to Santa's problem of chronic wasting disease. So Santa might first of all be interested to know just how much research is going on relevant to reindeer. So here you can see a very, very kind of normal search result on the left. If you were to search for reindeer across journal articles, clinical trials, preprints, and all of the associated literature, you essentially find that there's quite a lot of research just on reindeer in itself, 2,500 articles, and obviously nobody's going to go out there and read that. On the right, you will see that there are various what are called relevant concepts. So these are actually diseases which are more significant in the way that they're mentioned within that set of research for reindeer. And top of the pile is actually chronic wasting disease. There are in fact 84 articles relevant to chronic wasting disease and reindeer. And that is significant against all of the research that's going on for chronic wasting disease. So it makes it quite an interesting concept for reindeer. So immediately you've got an insight there in which you're seeing those diseases which are perhaps kind of hidden away beneath all of that research, which are being talked about alongside reindeer. So then the next thing you might be interested in is, okay, so if prion protein is the major cause of chronic wasting disease, how can we go out there and find all the evidence and the understanding of why that's the case? So if we look by doing what's called a causal search, we're looking to see all of the different examples in which prion protein regulates chronic wasting disease. We can see that there are various different results mentioned in many, many different ways. You've got PRMP gene there, talked about, lots of different synonyms and acronyms, etc., for both of these two concepts, and lots of ways in which they're shown to be related, which you can aggregate together into a single piece of evidence. So this becomes more useful now. We're starting to abstract all this information into something which is more organized, and so we can sort of get to the point and get to what we're looking for a bit quicker. So we can take this a bit more unspecifically. So we can look to see how PRMP has been shown to affect lots of different concepts. So we can see that in terms of other things that it's shown to regulate or increase or decrease, etc., that there are various different concepts that this particular protein has been shown to have a causal relationship with across all of the literature related to PRMP. So how can we take this a step further? How can we actually start to see some explainability behind prion protein and how this might impact something like one of the symptoms of chronic wasting disease. So we may have a piece of research which is talking about prion protein inducing the mitochondrial dysfunction, which would be some negative phenotype, which we want to avoid, obviously. Then you'd have another piece of research talking about that mitochondrial dysfunction leading to demyelination. And then subsequently, you might have another piece of research which is talking about demyelination of peripheral nerves leading to ataxia. So you connect together all of these different articles, all of these different cause and effect statements, and then you can start to understand new ways in which those particular concepts relate together, which gives you more information to delve into and to look into to see how these different concepts relate together. And so finally, you can take this one step further, where you can actually collect all of these cause and effect paths to provide even higher levels of abstraction. So here you can see a chart on the right, which is showing you the relationship between that prion protein and ataxia, which, as you will have seen on the first few slides, is one of the symptoms of chronic wasting disease. And at the bottom, you can see that this regulates the abnormality of the mitochondrial metabolism, peripheral demyelination to ataxia. So this is running left to right. This is essentially a chart that takes together all of these different cause and effect relationships. Each of the nodes, so each of these little boxes, are in fact sized by their enrichment. So the bigger they are, the more significant they are within that system, i.e. the more prominent that they've been mentioned against all of their other research across all of the other different contexts and all the ways that these particular concepts may be talked about. And so by taking all of this information together, we can start to understand the importance of these entities within complex systems, which otherwise would be very difficult to do from starting from kind of ground zero and just trying to understand how these different things relate together, particularly if they've never actually been directly mentioned together within the same articles, and where these kind of chains and cause and effect relationships are not easy to kind of tease out. So we can actually measure the overall significance between any of these different sources and targets. So here I've got a table that, apologies, there's lots of numbers here, so I'll try and talk you through what they actually mean. We're essentially looking at the relationship between PRNP and various different phenotypes and diseases. So the first example I've got on row 1 is ataxia, which you saw on the last slide. Now, if we're just looking for ataxia, we'll know that there's lots of research going on around ataxia. You've got around 47.1 thousand articles that just mention this particular phenotype. If you're looking just those articles that talk about ataxia against PRNP, you're down to 672. And there are in fact 92 sentences that mention those two concepts together, and 9 cause and effect relationships where in fact PRNP is being documented to have some effect on ataxia. And so we can actually measure the overall enrichment or significance of the link between PRNP and ataxia by using a clever play on an algorithm called the hypergeometric mean, which essentially measures the proportion of the connections represented in that subgraph between PRNP and ataxia against all of the other connections that each of those individual nodes has. So this gives you a p-value, and it gives you a way of showing the significance between any two concepts. And so knowing all of this data in terms of their level of study and their significance, we can then start to look at potentially other more interesting insights that perhaps use these stats in slightly different ways. So if we're looking for what I'd call a known, something that's very, very commonly mentioned alongside prion protein, you'd probably see that Scrapie is a good example of that. So you've got about 6.1 thousand articles that talk about this concept. Around half of those are directly mentioned alongside prion protein, and about a third of those are the sentences themselves are all related to PRNP as well, with 360 individual causal mentions. So everybody that's kind of in this particular field will obviously know that these two particular concepts are really closely related, and they'll probably have all of the understanding and detail behind why that is the case. But we might be more interested in those that just have this higher significance, this higher enrichment between the two of them, which might not be as obvious to the people that are kind of studying it, but through the ways that the concepts are regulated between these two, actually potentially are more significant. So we can see that the abnormality of the mitochondrial mechanism has actually got much higher p-values, much higher enrichment scores against PRMP, which potentially makes it a more interesting concept. There are, various articles that do talk about these two things, but by all means, it's not quite as kind of well known as perhaps alongside Scrapie. And so we can take this a couple of steps further. We can actually look to see, are there any articles out there that are barely researched together, where there is still significance between these two concepts, because they are essentially linking together different proteins or different chemicals between articles where these two perhaps haven't been mentioned together previously. So there are actually, against tuberous interstitial fibrosis, no sentences that are actually co-mentioned with prion protein, yet they are enriched at what we call depth 2 and depth 3, which are involves different steps of links between those two concepts. And in fact, something like non-communicating hydrocephalus has never been mentioned with PRMP before, but yet depth 2 is still significant, despite the fact that they've never appeared alongside each other in an article. So I find this particular information fascinating, and this is, I think, a very, very useful way to start to home in on potential hypotheses as to what might be promising drug targets or promising indications against targets, and the rationale between why they are so closely linked. Fortunately, Biorelate were able to supply Santa with all of the data on CWD and PRMP, so he could work his drug discovery magic. And we're told that Santa has found a cure, and he successfully tested this on his reindeer. So Christmas 2021 is saved. So fortunately, that's good news. Thanks very much.
Thanks, Dan. I think you did a fantastic job meeting your original aim of linking AI, neuroscience, and Christmas. So lovely example.
I don't think it's something I do so often.
So we're running slightly over, but I don't think that matters, because we're just officially having a break, and there are some questions in Q&A. So if people want to hang around and listen to these questions, please do. So let's start with Matthew Albrighton. Do we or will we see a growth of the evidence base for this and neuroscience per se on the Cochrane collaboration?
So the question was, sorry, it was, do we see a
growth of evidence base?
Yes. Yeah, so it's an interesting question. We're seeing a growth in evidence really everywhere. I don't think there's a single field where you're not seeing some level of exponential growth. And things like the Cochrane collaboration, they're a really interesting approach to actually trying to understand the states of research, and obviously based on a manual approach to doing it, where you're doing systematic reviews across a very carefully selected group of articles. I think that's obviously a very valid and high accuracy way of doing this. These types of tools are essentially meant to look in every single corner of research which has perhaps not been illuminated previously. So things like the Cochrane collaboration may miss because they go through a quite a stringent triage process to make sure that they're only looking at the articles that they believe to be important. But I mean, these particular tools are then looking at all the various different ways in which these things are mentioned and potentially finding data that has not been seen before. So yeah, I expect that this is something that will help, and it would certainly support things like systematic review and pragmatic review.
Thank you. And there's a question from Annette Dolphin. How do you weight the quality of research And do you exclude so-called predatory journals?
Yeah, so it's a very common question, actually. So what we try to do, and in all of the examples I've shown on these slides, this is essentially using all of the peer-reviewed research which is out there. So we haven't filtered out anything. But what we do have available to us is all of the knowledge of where each individual piece of evidence has come from, including its journal, its impact factor, its authors, everything, all the metadata around every particular piece of evidence. So if we are looking to only focus on, say, the natures of the world or the science journals of the world, then we can potentially just filter out everything and see what remains. But what we're kind of more keen to do at Biorelate is to let statistics solve these problems by essentially doing things like enrichment, which would hopefully illuminate the more interesting piece of information in most cases. But as I say, there is always the opportunity to filter out bits if you want to.
Okay, we probably only have time for one more question. So apologies to those who've put questions that won't have time to answer. Maybe if you put them in the chat, and maybe Dan will have time later today to type an answer. But to finish up this session, before we have a break, Levan Bakaria asks, how easy or hard would it be to do the same type of literature analysis on a field other than drug discovery, for example, neuroscience? So a few more questions is, what are the initial conditions that need to exist for your software to work? For example, would you first need to develop relevant libraries of ontologies for neuroscience?
Yeah. So that was actually the approach I was going to use to answering the first question. So essentially what you have to do for any of these concepts is you need to map them to a concept. So an ontology is the best way of representing all of these different concepts that are out there. So at present, what we use for ontologies are quite a wide range of different ontologies that capture every gene, as many chemicals, every disease, including all the neuroscience diseases, every phenotype, basically any concept that's likely to have been mentioned on something like PubMed or PubMed Central. So at the moment, we would already capture a very large range of these concepts, but then the ways that they're actually related together, we're only at the moment looking for changes in activity or abundance. So this might exclude certain types of relationships, which might be, I guess, slightly different within neuroscience research. But the software itself that we've developed that uses all of this this kind of structure is fairly malleable. So it's quite easy to extend and incorporate these other ways of looking at relationships. So this may be something that we think about expanding into the future. But at the present, this the initial kind of MVP for the platform is being used to support drug discovery research, and it's usually changes in abundance and activity that have the most value in those processes.